Empezamos.
Hi, everyone. Welcome to the second day of the Global Summer School 2021 Biomedicine and Biotechnology. This is hosted by the participating universities, ENES Juriquilla, UNAM México, UPIBI IPN México, Universidad del Piemonte Orientale, Novara, Italy, and Manchester Metropolitan United Kingdom. Good morning in Mexico, good afternoon in Italy and the UK. We will start our second day program with welcome you to this session. This session will be hosted by me. Well, I'll be the chair, and we'll see the work of several colleagues from UNAM. First of all, we will start with a welcome. The welcome already done. So we will start with a cultural session that will be tell us by Maria de la Teja Peñalosa. Mariana oversees the cultural and physical education area. She has a bachelor in administration, leisure, sport, and physical activity, and is currently enrolled in the master degree on project management. Her activity with the ENES include coordination, physical and training activities, and cultural events, such as the annual welcome event for the new students. She's also in charge of the volunteer program of NAM and PERA program. The purpose of this program is to help middle and high school students that need emotional and educational support. Mariana's work was especially valuable during the lockdown as a result of COVID pandemic, when students needed encouragement, support, and a friendly face to continue working online and in isolation. We welcome to Mariana de la Teja Peñalosa, who will give us the first session in our program, which is the cultural session. Many thanks, Mariana, and welcome to the Global Summer.
Hey, thank you so much. Can I be able to share my screen, please? Thank you. Thank you so much, Luis. And yeah, I will be talking about what to expect if you come to Querétaro for the Global Summer course. And I will start with something that is really obvious, but I have to say it. And I don't know if you can see my screen now, but if you come to Mexico, to Juriquilla, that is in Querétaro, you will find Mexicans. I know it's pretty obvious, but you need to understand that I'm pretty proud to be a Mexican. We are really friendly people. Not everyone speaks in English. Our native language is Spanish, but everyone will try to understand you and to be your friend. You will have so much friends of Mexico, because just in 1 hour, you will be in their houses or eating with them in a tour, in a party. So I will have to start with that Mexicans are really nice persons and we're really cool. So where is Querétaro located? The official name in Querétaro is Santiago Querétaro, but we name only Querétaro, which is the same name as the state city, as the capital. And we have nearly 1 million persons, 1 million people that make the probably 24th largest city in Mexico by population. You know, Mexico as a country, it's pretty big. This is the number in square kilometers. So this is the map of all Mexico. You can see it's a really big continent. And Querétaro is located right here in the Mexican plateau. And it's about 210 kilometers from Mexico City. It's pretty close to Mexico City. And it's close to another really nice states like Morelia, San Luis Potosí, León. So if you come here, you can go to another states pretty close in car. You don't have to go to airplane. Because, for example, all the north part, you have to go to with an airplane because it's really far away from the Querétaro and from Mexico City. What to do in Querétaro? We have a lot of activities, of cultural activities. And the first thing you have to go in Querétaro, obviously, is go to the historic center. And there are about 1,000 of monuments through the historic center. Most of them date of the 1700s, when the city starts in living, and it's recognized by the UNESCO of World Heritage Sites because it's really conserved. It's really beautiful. It's really clean, a really clean city. And you can do a lot of activities in the downtown, in the center. You can go to restaurants, you can go to bars, you can go to dance in some of the plazas. We have a lot of restaurants that are in balconies, so you can have a really nice view. So you can do a lot of things there. You can always go to the aqueduct that is about 2 kilometers long, and about 30 meters looming in height. And this is kind of a Roman structure. It's where we used to put the water there, obviously, so they transport all the water to all the city. And you can take really nice pictures there, but actually this type of pictures, you can see it in the Mirador of the Arcos, that it's a lookout point in the eastern edge of the center. And in the shore of this aqueduct, you can find a lot of restaurants. So a good plan is go first in the historical center and then go to the aqueduct to have dinner, just to see this incredible aqueduct. We have a lot of museums too, and you can see there are gorgeous museums in the buildings. Per se, there are beautiful museums, beautiful buildings, and we have a lot of type of museums. We can have the art, contemporary art, or we can have the traditional ones. The first one, it's actually not a museum, it's El Cerro de las Campanas, that you can visit. It's a really important part of our history. Querétaro, it's really important in our history, in the independence and in the revolution, in the Mexican Revolution. So you can visit all these buildings in the historical center. You can go in this type of tours. You have the traditional one that is in the left, or you can have the bus, like the typical bus of 2 floors, and have these like tours to the aqueduct, to see the museums, to see the monuments. Another thing that we have is churches. And in the historical center, you can have 1 church every block. So you can visit them, and they are really, really nice churches. And these type of tours, I recommend you because you can see everything like in a really fast way. And then if you notice something that you really like, you can go by feet and visit it in more time in your visit. If you come to Mexico, to be honest with you, you have to go to the markets. Here we have the Mercado de la Cruz, that is the biggest one, but we have another small ones all around Querétaro, but it's a must to go to a market in Mexico. It's pretty nearly to the one of Italy, but with our local products, obviously. And we'll see all the colors and the fruits, the veggies. You can find piñatas, the famous piñatas that we break in our birthdays and in Christmas. You will find all our local products, and the best thing is that you will find it cheaper than in any other place. Wait for me just a little bit. They're saying me that I'm having a little bit problems of internet. I'm sorry for that. I will try to go slower. So if it's cutting, it will be like understanding better. I'm sorry for that. Another thing to do in a market is the food. You can find the food there. So obviously these locals of food, they are buying all the things in the market, and you can eat in the market with the food just about one step from you. So everything is really fresh and everything is really delicious. So it's a must to go to a market, and Mercado de la Cruz is the biggest one in Querétaro, so I recommend you to go there. Street food in Mexico, it's awesome. In the cultural session in the afternoon, my other partner, it will talk about that. But right now, I will talk just about a little bit of the street food. Just be worried because foreign people sometimes get a little bit sick with the street food in Mexico. But if you eat 1 or 2 times with medicine to prevent that, you will be fine. Don't worry about that. Here in Querétaro, we have garbanzas, we have corn food, we have gorditas of maíz quebrado, and obviously all the type of tacos you can imagine in Mexico. It's obviously another must in when you come in here in Mexico. Our gastronomy, it's amazing. We have a lot of type of food, even if you don't eat meat or if you don't like a certain type of food, we have a huge amount of type of Mexican food. So restaurants are pretty good. I'm not saying that they're not, and they're really, like, we have a lot of type of restaurants, Mexican, Asians, Italians, French, a lot of type of restaurants, but the street food in Mexico, it's beautiful, and you will find a lot of type. Another recommendation is my personal recommendation is go to Lucha Libre. These wrestlers in Mexico is super traditional. It's a cultural thing. And we have 2 type of wrestlers. One are the técnicos, that are the good guys. And the other ones are the rudos, that are the bad boys. So the good ones are these wrestlers that are super athletic, and they jump from the 3rd row, can have chains and a lot of spectacular things. And the other ones, los rudos, the bad ones, they are always cheating, and they have a little bit more big. They're not that athletics, but we have a referee, an arena, that is the ring, and a lot of really good snacks, and particularly from the Lucha Libre, like, for example, los cueritos, that are the skin of the pork, but not like the chicharrón, more like a yellow. It seems kind of gross, but it's really good when you put lime and salt. And you have to go to a Lucha Libre show, because, for example, you can find old lady, like a 80-old lady outside of the arena, waiting for the ticket and waiting for get inside. And she will be the most careful and the most such a lovely person. And when a Mexican go inside a Lucha Libre, you will hear them shouting a lot of bad words. It doesn't matter if you have 80 years, 3 years, 15 years, everyone is gonna be shouting. And I recommend you to go with another Mexican, because sometimes it's like, oh my God, what is happening here? This is a lot of crowd, and everyone is shouting. Everyone is like trying to fight with the other band. Nothing like aggressive, but it seems to be like that. In Querétaro, we have a lot of pueblos mágicos, called in Spanish. They are called Magical Town Program, and it's an initiative from the Secretary of Tourism with support for federal agencies to promote a series of towns around the country that offer visitors special experiences. It can be folklore, historical relevance, cuisines, arts, great hospitality, ecotourism, I don't know, like rural areas, etc. And in Querétaro, we have the fortune that we have 6 of the 132 magical towns, and they're really close to the center of Querétaro. For example, this picture is from Peña de Bernal. It's one of the most beautiful towns near Querétaro. It's about 45 minutes from the historical center. And you will find this monolith that is one of the biggest in the world. It's 433 meters of tall. You can climb to the middle part. It's a hike that you can go in there. And you can actually go to the top, but you have to have the equipment because you have to climb. And it's better if you go with a nice company and you climb in this section because it's kind of a difficult part to climb. But as you can see, you can sit down in a restaurant and have this awesome view, or you can go to the monolith and have this really good hike. And just near this town, the La Sierra Gorda, you can see how it's starting. So we are surrounded by mountains, and it's really good weather because of that in the center of Querétaro, in Santiago de Querétaro. In summer, it's an average of 15 to 28 Celsius degrees. So it's really good weather. In summer, sometimes it's raining. Right now we have a rainy weather, but it's still not that cold. And you can make a lot of activities even if the weather is not that good. These are the other 6 or 5 towns that are near center of Querétaro. That is Tequisquiapan, Cadereyta, Amealco, Jalpan de Serra, Cadereyta, and San Joaquin. And you can see they're really beautiful towns. In Amealco, actually, we have this doll that is called Lele, that I will explain you a little bit more after. And for example, in Jalpan, that is in La Sierra Gorda de Querétaro, you can have a lot of ecotourism and hikes and a lot of churches, beautiful churches that are called misiones, and that San Fran, well, whatever. Then we have the vineyards that they are really famous here in Querétaro. A lot of Mexicans come here to Querétaro because we have these gorgeous vineyards. We have a lot of them. The most famous is called La Redonda, but you can find ones or really, really big ones, and you can have a really good breakfast or a dinner there, or a lunch, whatever you want. And the good thing about these vineyards, they are really close to the magical towns, to los pueblos mágicos. So you can have a plan to go to Peña de Bernal, for example, and go to one of these vineyards. So you can do 2 things in one day, and it's gonna be really close to each other. And you can have, almost all of them have tours, so you can have like a sommelier experience of the wine. And you can find really big restaurants and local wine, obviously, so you can try them. Always in near Querétaro, in Corregidora, that is like 7 kilometers from the historical center, we have this pyramid called El Cerrito. And this pyramid, they don't know exactly what type of civilization go with this pyramid. They're supposed to have Teotihuacanos, Toltecas, Chichimecas, Purépechas, Otomís, and Chupicuaros. I'm sorry if that sounds kind of weird. It's not Spanish, actually, it's from our Asian civilizations. The most famous, obviously, is Teotihuacan or Chichen Itza, and the Mayans and the Aztecs. But in Querétaro, we have a lot of that type of culture too. And this pyramid, if you want to see like a little bit of how was our civilization, it's only 7 kilometers from the historical center. So it's another recommendation. Another thing I want to say to you is that the museums, the tours, this type of cultural sessions, they're really cheap. And the museums are not more than $3, €3. And to go to El Cerrito or to enter there is almost the same. And street food is super cheap. You can actually eat, for example, an elote, that is the corn with mayonnaise and chili and lime for maybe $1 from $1 or €1. So they're really cheap in Mexico to do all these activities. The ecotourism here in Querétaro is really nice too. This picture is of the Mirador de Cuatro Palos. This in the Sierra Gorda, where I was telling you about Jalpan, for example. And this region has an ecological region called La Biosfera. I forget it how it's called. The Sierra Gorda Biosphere that was established here in Mexico, and it's about 400,000 hectares. So that means it's a lot of the territory of Querétaro. And it's a reserve because we have a lot of species and a lot of beautiful things like waterfalls and that type of things here. And it's located in La Sierra Marental Mountain. And it's a series of mountains in Mexico. This is part of that one. It's a huge mountain, so, well, a series of mountains. Like you can see here, like I put a lot of type of ecotourism here in Querétaro. The biggest one that is in the right part of the presentation, it's called El Cañón del Paraíso, the Paradise Canyon. And you can go to have a hike there. It's not, it's really easy hike. And you can go in the center of the canyon. Sometimes when it's raining, it's a little bit higher, the water, but not that bad. You can swim in some of the pools and you can continue walking. It's a beautiful place to have hikes. The one in the top part, it's Puente de Dios, near Chuveje too, that is a waterfall. And you can see the color of the water is like really blue, and it's a really good place to go and swim and have a little bit of hikes. That's in the Sierra Gorda. And in the down part, this is called the Cimatario. It's really close to the center of Querétaro. The other two are like 2 hours or 3 hours from the center, but the Cimatario is really close to the historical center. And you can go walk there and have really good views from Querétaro. And this is the campus of the NES Juriquilla. Juriquilla, it's a really small part of all the Santiago de Querétaro. That's why I'm putting you this. This is the campus we are trying to build. Right now, we have, I think, 3 buildings in construct, but this is how it's gonna be if you come here to Querétaro with us. I was telling you in Amealco, we have this type of doll. It's called Lele. It's a really traditional doll here in Querétaro. You will find it almost in every part of Querétaro. It's a good thing to get back to your native country, because it's a really cultural thing for us. And you will find it in a lot of colors, a lot of dresses, and it's really traditional from here. It's really beautiful, and you will find it for really cheap prices. So if you come here, you have to buy them from the local persons. It's a must too. And because of, I know we cannot do a lot of activities. To be honest, it's kind of weird for me to have this type of activities. Because I'm talking to a screen. I would love you to come here and we can have all these cultural things, and I can show you like all the things I'm telling you like in life. But if you want to participate in an activity, we will have this picture, this image in the Facebook in a link. You can download it and you can decorate all the doll, and you can send it to my email, and we will try to recollect all these dolls and put it in the official Facebook and Twitter and Instagram from the NS Curiquilla. So if you want to participate, it will be so beautiful to have those from Italy, from Manchester, and from Mexico City. It will be awesome you to participate in the activity. Hopefully next year, if the pandemic is not this bad, we can actually go and buy some of these little dolls. And that's it for me, Luis. Thank you so much for your participation, and thank you for inviting me.
Thank you, Mariana. Really, really nice description of Querétaro. It is a lovely city. I hope everyone can go and visit soon. It is really, really nice city. Well, then we move to the academic part of our Global Summer School. We have the first talk. It is hosted by Dr. Octavio Diaz. The name of the talk is Research in Orthotics, Prosthetics, and Rehabilitation Technology. Dr. Octavio Diaz Hernandez received his bachelor's degree in mechatronic engineering from UNAM in 2007. He also obtained a master doctoral degree in mechanical engineering, both in UNAM. He has been professor in the School of Engineering and the School of Medicine. His research involved robotic systems, automation, mechatronic design, teleoperation, electronic instrumentation, and mechatronical system applied to medical rehabilitation. He has published articles on his research in national and international conference and international peer-reviewed journals. He had direct theses in different areas of engineering and has participated as examiner in multiple exams at undergraduate and graduate levels. Since October 2020, he has been a full-time professor at UNAM Juriquilla, assigned to the electronic area in the bachelor program of orthosis and prosthesis. We're welcome to Dr. Octavio Diaz Hernandez, and welcome and thanks for assisting to this Global Summer School.
Yeah, thank you. Thank you all for the invitation. I will share a screen for the presentation. Give me a second. Well, thank you for the invitation before all. And what will be about us if we are not with the team? Yeah, we are a part of the biggest team, and well, I have the assignment to talk about a little bit, just a little bit from the research we're doing in this area, okay? And I will show you, first of all, the problem. The problem in Mexico and around the world is that there are patients with amputation. Here in Mexico, about 900,000 people with amputation, which is bad because it affects the daily basis activities from the patients, the work, and socially bad for them. Then the causes from the amputation are around about these ones: trauma, congenital tumor, and some diseases just like diabetes. And in Mexico, diabetes is very large amount of people, sadly. And the problem also is that it's a low number of professionals in the area of orthosis and prosthesis. Well, but what is that? What is orthosis? Well, it's a device. It is applied externally and is used mainly to recover functions, to give the person who has lost a function, which can be muscular and skeletal problems, which mean in the bones, articulations, and is trying to regain the function of that. And the main difference with prosthesis is the prosthesis is replacing some part which is lost, just like a finger, a hand, a limb. Okay, and there are a lot of devices around that for the orthosis and prosthesis. But the rehabilitation, which is another area very intertwined, is the regaining daily life functions for the people. Yeah, you have orthosis and prosthesis, but you have to regain those abilities which has been lost, such as walk, such as work, and other activities. Okay, rehabilitation. And well, I have this task to tell you about the research. And as you can see, there is some places you can be working in. For example, in the NS, yes, there is a building, a little part of what is building, what has been built. But if you come, you will be seeing some larger buildings already built. Okay, so as we can see here, there is some prosthesis, limb prosthesis, and some from the thoracic or upper limb as well. Another thing is that you can be working in the biomechanical lab, which is a great place to be studying and making research. Okay, I will talk about that, something about that later. Okay, well, the team, Dr. Jesus Manuel Dorador is mechanical engineer, and he has his PhD from the United Kingdom, and is our head leader in design and innovation. In, well, some of the research, not all, is about a self-adjustable socket, which helps to the people who has a transfemoral prosthesis to be more comfortable. Okay, which is just one of the projects we are working with him. Another aspect of this adaptation of orthosis and prosthesis is physiotherapy. Yes, the doctor Igor Salinas is a physician in physiotherapy, and is in charge of evaluation, yes, in all the process of the adaptation of for a patient with orthosis or prosthesis. Yes, he will be in rehabilitation, physiotherapy, okay, which is some different about our partner, Alejandra Santos, which is bachelor in orthosis and prosthetics from El Salvador. It's a school in Latin America. And she has a master in biomechanic clinical assessment from Spain. So she is studying the biomechanics to the adaptation. He will be in the clinical assessment of the patients that will be using these kind of devices. Okay, and let me talk about something from biomechanical, which will be all intertwined with the biomechanical, as you will see. Well, as another partner of our team is the PhD and Dr. Raide Gonzalez, which is from Cuba, and is studying, analyzing through computer software all the biomechanical aspects of the gait which is you've been working, and the persons with the amputation. Okay, and there's so much to do about that with the cameras and specialized software. Well, in another aspect of this bachelor and the research, will be needing technology, of course. And Asael Solalpa is a mechanical engineer, and he has a master in science of technology, and he's working with cutting edge machines, just like the manufacturing, additive manufacturing, or 3D printing, and CAD/CAM, which means that computer-aided design, computer-aided manufacture, and we're going to be able to use cameras to digitalize images of patients and devices as well. So in here we can see a machine which is very, very specific to males and is very useful. It has 5 axes of to be working with the devices we will be designing and manufacturing. As you can see also, there is an orthosis example in the software. As you can see, is the computer-aided design, the CAD. Okay, here as well, it's a socket from a prosthesis, which has been modeled in this kind of software. Well, about myself, well, as I've been introduced, I am a mechatronical engineer, also with a doctorate. And well, I've been working with rehabilitation for another diseases, just like the stroke, and the neural rehabilitation. And it is my interest to introduce a new line of research, which is soft actuators, that has in the main basis a silicon, which is a material, very soft material, elastic material. Yes, this is an example that has been applied to orthosis. Yes, and as well, is my interest with the team to work in this line to apply more and more these kind of materials. These materials has very singular properties, just like to be very similar to the living tissue, such as human muscle. So as you can see, it will be in the hand, in elbow, ankle. So also will be built to aid gait, as you can see in this image, the cycle of gait, and will be useful for the analysis as well. Also for the rehabilitation or adaptation to the prosthesis and orthosis. Well, if you want to come, of course, you will be welcome. As you can see, there are students participating. For example, master degree and bachelor degree are using many sensors. Some sensors we have instrumentation in the biomechanical lab. So you will be very, very welcome. Well, let me finish with just a little bit of conclusion. This is a building which is in progress, and if you come, we hope will be more and more terminated. Finished. Well, the team, as you can see, is working with a lot of lines, and this main star is the biomechanical. Okay, and there is so much projects that you can participate. Not only the ones I told here will be larger speech here to talk about details of all these research. So with just a little taste for you to be joyful to come and to participate. Well, and thank you for the attention, and well, some of the team I was glad to introduce. So you will be welcome to participate, and thank you for inviting me to this chat. Thank you.
Okay, many thanks, Dr. Diaz. A really interesting talk about this important issue. I don't know, we have some questions. Let me check. In the meantime, could you tell us more about the program, the graduate program at your department or your university, or if you can give us more information? But if someone is hearing us and wanted to apply or is interested in these topics, that would be really helpful. You can give us a little bit more of information about it.
Of course. I had no a slide for it, but I will talk about the main things. Just like this is a bachelor for persons who has been left college, and they have to be evaluated, and they have to pass some exams, and they can be from two areas. Very interesting because it's an hybrid area. What I mean? I mean they can be just engineering. Yes, this is a line where you can be coming from, or even a physician or a physiotherapist, and another biological area. So this transdisciplinary bachelor, I want to tell you, well, you will be studying things about engineering and from medicine sciences. So what you can study here is, as you can see, clinical assessment, which is very important, and the different aspects of the technology for manufacturing, as we can see here. Yes, the team is built about engineers, yes, in the mainly basis, and physicians, okay, and Alejandra is our only bachelor in this area. So very important to be clear that is a mixed and hybrid bachelor. You will be seeing the two things, the two branches of the sciences, medicine and engineering, and the subjects of the study plan, okay, will be around that. Also, UNAM, this character is very interesting, that gives socio-humanistic subjects as well. Thank you.
Okay, no, thank you so much. It was really interesting. You know, that is a multidisciplinary program, and physicians are people from the biological science are welcome to apply. Hopefully, some people is seeing us right now, and he's interested in the program. Many thanks, Dr. Diaz. Okay, we move on to our second talk. The second talk of our day is, the name of talk is Finding Ancient Pathogens Through Paleontologenomics. This talk will be hosted by Dr. Maria Avila Arcos. Dr. Maria Avila Arcos obtained her bachelor's degree in genomic science from UNAM in 2009. Soon after that, she moved to Copenhagen, Denmark, to study a PhD specializing in paleontogenomics at the Center of Geogenetics under supervision of Tom Gilbert. She obtained her PhD diploma in 2013 and stayed an extra year as postdoc. Before joining the International Laboratory of Human Genome Research as a junior faculty, she was a postdoc in Carlos Bustamante groups at the Stanford University for almost 2 years. Her research interests revolve around the study of human population, past and present, to learn about the process that generate the genetic makeup of a group of humans through time, with a particular interest in understanding population from Mexico and Latin America. Her work combines the analysis of genetic driving from living and past population to understand cultural, demographic, and adaptive processes that have shaped the observed patterns of diversity in such a region through time. We welcome Dr. Maria Avila Arcos.
Hello, everyone. Thank you very much. I'll start sharing my screen, and then I'll do some quick tweaks. 1 second. I will, okay. Can you see my screen?
Yeah, if you just want to present it, put it in
okay.
there.
Let me see if this works.
Yes.
Okay, hi everyone. It's a huge pleasure to be here today. I'm gonna tell you a story today. I am a PI at the International Laboratory for Human Genome Research, and I want to apologize in advance if my dogs bark. They are around, so every time some dog passes by, they bark pretty loud, so I apologize in advance. So this story I'm going to tell you today, I chose it because it's going to tell you about pathogens, and I think this is very relevant in the times we're living in, but how we are using or leveraging the information from the past to understand the biology and the evolution of pathogens with a very clear temporal framework. And I also chose this research because it shows the interdisciplinary research we do at this in my group. And I just want to tell you a small warning. I show in some slides some skeletal remains to make some points. So if you are sensitive to this, I guess you are welcome to ignore this talk. Just want to make this this warning first. So to start, as I mentioned, I am at the International Laboratory for Human Genome Research, or LIG, at Juriquilla. This is our building. It's relatively new, around 6 years old. And right now we are about 8 young faculty doing research in very different areas, from cancer, bioinformatics to genome dynamic evolution, and my group where we do population and evolutionary genomics. So this is, I wanted to show you a picture of how it looks. It's a very beautiful building with a very beautiful surrounding. So to start, to jump in directly in this story I'll tell you today, I show you here a graph of the population in the Central Valley of Mexico, right at the point of European colonization. So what we see is that soon after the arrival of Europeans, because of large epidemic outbreaks, the population declined drastically. So it says that around 90% of the population died, mostly because of the newly introduced pathogens, and also because of massacres, also because of marginalization and other social aspects, but pathogens played a very important role. So here I'm showing you, for example, smallpox alone caused around 8 million deaths very soon after the arrival of Europeans. And also there are two outbreaks here that the name is Cocolistli, which means pest in Nahuatl. That's the local language of the Central Valley in that time. And you'll see the number of deaths caused by this unknown pathogen. We don't know much about this pathogen. It's very, very large. There's been a speculation about what this pathogen was. Here are some depictions of what it caused to patients, some rashes and some pustules in the body, intense bleeding. It looked like an Ebola-like disease, but we don't really know what it is. So of course, one way to learn about what these pathogens were or why they were so deadly is to study the DNA. So take COVID, for example, today, we are learning about these new variants. We are learning about how it affects the host based on the genomic sequence of the viruses. So it's to study the DNA, it's a key aspect to understand the biology and evolution of these pathogens. So that's what we attempt to do, but with pathogens in the past. So to achieve this goal, we can take DNA from skeletal remains. There are different sources, and there are two main categories of the sources we can take. So one is when we have clear bone lesions that pathogens leave. For example, here I'm showing you an example of Trapanomatosis, which is caused by Treponema pallidum, so that's syphilis. And this is a lesion caused by TB, like tuberculosis in the vertebrae. And these are very distinct lesions that we could, in principle, sample directly from the bone and try to recover the DNA of that pathogen. However, if you think a bit more, in order to develop these lesions, these are pathogens that had to be in the host for years, if not decades, right? So we don't really know if the person died of this pathogenic infection or died of something else, because apparently their system was resistant enough to leave these pathogens stay for so long that they could leave these lesions. But if you die in an epidemic outbreak and die in the matter of days or weeks or months even, maybe these pathogens won't leave a lesion in the bones. So what we do is to take the archaeological context that suggests that there was an epidemic and sample the teeth. And why the teeth? It's because the teeth, the root of the teeth has blood vessels passing through. And if you have sepsis, for example, you assume that you would have enough pathogens in your bloodstream to be able to recover it from the root of the teeth. So here are some examples of pathogens that have been retrieved using this strategy, and this is the strategy we followed in our lab. So the story I'm telling you today, it's how we screen for pathogens in some individuals from the archaeological record from the colonial period in Mexico City. We studied two sets of archaeological remains. One was from a hospital called the Hospital San José de los Naturales. This is the location in Mexico City. And the other one is from Coyoacán, church and colonial church. Actually, this was one of the first churches in Mexico City or in the New Spain, and we sampled some individuals there. So we chose the hospital for obvious reasons, the graveyard of the hospital and the church. Also, there were some records that the population was very dense in this area of the city, so we could expect maybe to find some pathogens there. So something I've noticed is that in the hospital, there were some individuals, even though this hospital was the first hospital to be created only for the native or the indigenous population. That's why it's called of naturales. That's the term that was used to refer to the indigenous population. Some archaeologists, one of them, my colleague, Julie West, identified that some of the individuals from this collection had some distinctive teeth marks that were suggestive of an African ancestry of some of the individuals. So this was something of notice, and I'll go back to this in a minute. And this is also a project that I carry out with the collaboration of Dr. Marcela Sandoval. So this is just to show you these distinctive teeth marks that are particular of Africa that caught our attention from this collection. You'll see why this is relevant. Next, so we wanted to extract the ancient DNA from these teeth. Something very important to know is that extracting DNA from old remains is very, very challenging. We face a number of challenges. One of them is that with time, the DNA starts to degrade, so it's very fragmented. We also have something that we like to call damage. Basically, it's a modification in the DNA that when we sequence this DNA, when we recover the letters in the DNA, we can be confused by these changes from cytosine to uracil. And this is something that accumulates with time as well. So we have to control for this, but it also gives us some hints that when we have the ancient DNA and we sequence it, if we see these changes, this is telling us that it's actual ancient and not modern contamination, which brings me to the next point. Contamination is a big problem in ancient DNA. What I mean by this is when we extract DNA from, let's say, a tooth root, most of the DNA that we recover is actually environmental DNA. These are the microorganisms that have become part of the sample as well. Like we can have all sorts of bacteria, archaea, or even plant DNA, fungi DNA. We're left with a very small fraction of what we call endogenous DNA. If we're not careful, we can also contaminate our sample with DNA floating around in the lab. And that's why we have to have extreme precautions of when we handle ancient samples for DNA extraction. So ancient DNA is commonly referred to as a needle in a haystack problem, where we have really to recover like a minimum fraction. And when we are interested in pathogen DNA, it's even less, right? It's not even the endogenous from the human bone, it's the pathogen there. So it's very challenging to do this. In my lab, we have what we call the Human Palogenomics Lab. It's in an independent building from the main lab, so we avoid contamination, And we have to enter this lab covered in bodysuits with face masks, gloves, boots, all sorts of protective body gear. And it's compartmentalized. Here are two compartments where we process the samples on one side and we extract the DNA in another side. And we take a lot of precautions to avoid contaminating the DNA ourselves, the samples with our DNA, sorry. Okay, so this is the space that we use. And going back to how we do this processing, I will try to simplify all this, but let's say we start with the teeth. We do all the lab processing to generate the data. So we prepare the DNA in a way that we put it in a machine and we generate a lot of readouts or reads of sequences. And then we can break down the sequences we get between the human fraction and the pathogenic fraction using bioinformatics. And we use the human fraction to do human-specific analysis. And all the pathogen DNA fraction, we can do some taxonomic profiling. And then in this taxonomic profiling, we can identify pathogens. So this is what we do here. But as I mentioned, because we have so little of this DNA, what we do is to try to fish it out with some techniques, some molecular approaches, and we basically fish out the pathogen DNA. And we sequence again that pathogen DNA. And we do some extra bioinformatic analysis. We validate that it's actually ancient and not modern contamination. all sorts of QC. And one of the main aspects that we want to get from this is a phylogenetic analysis, right? How these ancient strains correlate with present-day strains or other modern, other ancient strains if available, and try to make evolutionary inferences from this. So this is the process of how we do this analysis. As I mentioned, this is the strategy. We try to fish out the pathogen DNA, and in this particular project I'm telling you today, what we were interested in fishing out viral DNA. So we were fishing out all families of viruses, but I will focus on the hepatitis and a parvovirus called V19B. So we were able to reconstruct 4 ancient viral genomes, 3 for the V19B parvovirus. And here I'm showing you the depths. This is how many times we read the genome. And a coverage plot. This is a linear genome, linear DNA genome. And we recovered 1 genome from hepatitis B. This is also showing you the coverage of the genome. If there are questions, I can tell you more about these interesting patterns of coverage that we found. So 4 ancient viral genomes. Let me tell you quickly about this V19B infection. This is parvovirus because you maybe have not heard about this before. So it infects the precursors of the erythroid lineage. It's very common, actually. Many people have had this disease without even knowing it. It's transmitted through the respiratory route. It can be found in several tissues. In kids, cause something called the fifth disease. It's a rash on the cheeks. It can be lethal if it affects pregnant women, causing something called a drops fetalis, and also can be fatal when it's found with immunosuppression, anemia, malaria, and hemorrhages or intense bleeding in patients. So just keep this in mind for a little bit. So what we did with these genomes was to place them in phylogenetic trees of worldwide variation. So this parvovirus, V19V, falls within genotype 3. What does this mean? So if we look at this is a phylogenetic tree, we have 3 main genotypes, 3 main clusters. Genotype 1 is globally distributed. You find it everywhere. So the names in black are sequences from the present, in blue from the past, and pink also from the past, and in red is our study. Genotype 2, it's found mostly in Europe and thought to be maybe out of circulation. It's only found in elder populations and in some ancient individuals as well. And genotype 3, it's very interesting. It's found mainly in Africa or African descent people. And our individuals, the 3 individuals 2 from the hospital and 1 from Coyoacán, fall within this cluster of Africa. Similarly, the hepatitis B genome, sorry, that's my dog. The hepatitis B genome falls within genotype A. I'm sorry. Hey, sorry about that. Let me grab my dog. Yeah. So it falls within genotype A variation, and what does this means is that it's also a clade. The genotype A is also found in Africa, in South Africa, and our individual that has it is it's also lightly African. So we could, of course, take this human fraction and try to infer their genetic ancestry. So we do something called PCA. We can make a plot where we tear apart populations with a reference panel. So here I have Africans, Asians, Europeans, and Mexicans and Peruvians. And our individuals that were infected with V19B fall within the African cluster and 1 in the Mexico cluster. And also individuals infected with hepatitis B falls in this African cluster. So this individual, one of these individuals with parvovirus, it was a male adult, 20 to 35 years old, with something called porotic hyperostosis, criba orbitalia, and both are associated to anemia, which is very interesting. So in summary of this, with what we gathered from this is that this is the first observation of this parvovirus in ancient samples, and the first one that we observed in Mexico, and that the chain of transmissions of this individual had to involve very likely an African individual. And we could complement this information determining the geographical origins of the individuals, because we were that genetically they were African, but we weren't sure if they were born in Africa. To do that, we use a strategy that is called the Strontium isotope radioanalysis. Basically, what we show here is comparing the strontium isotope mark that you have in your teeth is reflective of where you were born, and the one you have in the bone is more reflective of where you spent your last years. So I'm showing you these patterns here of these two individuals. If we look at the tooth, they have values similar to those observed in West Africa. And if we look at bone, we look an intermediate value between that observed in Mexico City Valley and in Africa. So with this, we can confidently say that they were born in Africa. And this is, of course, a legacy of the transatlantic slave trade into New Spain. We know that around 250,000 people were forcibly taken from Africa to Mexico or to New Spain there during the transatlantic slave trade between the 16th and 18th centuries. And there were actually, at a point in our history, mostly in the early days, this is African populations, European, African, native, and mixed populations. The African population outnumbered by many fold the European population. So in the very first, in the first years of the colony, the African population was larger than the European population, right? So we have a legacy of that today in our genomes. So to wrap up this story, I want you to have these take-home messages. We can recover ancient pathogens from teeth of infected individuals from the past. With the strategies I show you, we were able to reconstruct 4 ancient genomes, 3 from parvovirus and 1 from hepatitis B, using these methods I told you about. When analyzing the phylogenetic relationships, they fall within African genetic variation. And this was consistent with the genetic ancestry of 3 of the hosts. One of the hosts, the one with native or admixed ancestry, had also parvovirus, and we suggest that the transmission had to involve people of different ancestries. We use these strategies with the strontium radioanalysis to support the geographical origin of the individuals, and we assert that they were very likely introduced to Mexico from Africa during the transatlantic slave trade. So with that, I'd like to end and thank key people in this project, my PhD student Miriam Bravo, my colleague Daniel Blanco, and an undergraduate student Axel Guzman Solis. You can see many people have been involved from different institutions, different funding agencies, INA, the Anthropology and History Institute in Mexico, and a lot of people actually. So with that, I would like to end and thank you for listening to me today. I'm happy to take questions if there are any. Thank you.
Many thanks, Dr. Avila. Really interesting talk. Let's see if we have any questions. I have a question. What is the successful rate to sequence or obtain a positive match for the DNA in this? You tell that was really, really difficult, but there is a percentage or something?
Yeah. So, well, in this example, we sequenced, I think there were 28 individuals, and we got these viral hits in 4. And we had to apply these strategies to get more sequence. So, because of the context, including which was a hospital, in one of the cases, it was very likely we would find a pathogen in there. So that's like kind of a success rate. But what I can tell you also is when we extract DNA from, well, most of the samples I've analyzed, the amount of endogenous, that's like human DNA, the fraction is very often more than 1% of DNA. So 99% is bacterial and environmental DNA. So those are 2 numbers we have to deal with in the field.
Okay, thanks so much. I think we don't have more questions. We really thank you again for your participation in the Global Summer School. And thank you so much.
Thank you.
Okay, following the program, we have a break. We finished the first section of our day, and we will continue in 15 minutes.